It's one of the top companies in genomics worldwide. And today, I'm very happy to have Melina Yakashvir as an account manager for Novogene Europe, and Arman Mehra, who is a product marketing manager, also for Novogene Europe. And they have prepared a very nice webinar for us. A quick reminder, we are Recrop. We are a cost action related to reproductive enhancement of crop resilience to extreme conditions, climate conditions. And one of the key aspects of our cost action is to explore how plants respond to different stressful environments. And one of these aspects is epigenetics, genomics, epigenomics. And I think we will hear a lot about these things from Melina and Arman. If you have any questions, you can note them down, and we will have a Q&A in the end. Or you can also write your questions in the chat. And I think Melina and Arman will pick up some questions and they will answer. Yeah, the floor is yours. Thank you so much. And thanks everybody for being here. Don't forget to push, it's recording already. Super. Okay, good.
Yes, thank you so much. Welcome everyone. Thanks for all joining the webinar. And I would additionally like to personally thank Akis for inviting us today, and our extended gratitude to Recrop and Cost for allowing this webinar to happen. For those who don't know me, as Akis has already introduced me, I'm Melina, your account manager at Novogene, and my colleague here who's with me today, Arman, is our product marketing manager. And we will be diving into the potential of next generation sequencing and exploring the innovations, applications, and giving you an insight to what you can really achieve. So as a brief overview, I would like to give you some background on Novogene, and then our latest updates, as well as then I will pass it on further to Arman, who will give you more of an in-depth on the next generation sequencing platforms and applications, as well as the workflow to obtain your data. So for those who have never heard of Novogene, we are an international company starting off initially as a bioinformatics team who wanted to improve the sequencing data quality that we were obtaining and analyzing. Therefore, we evolved into a sequencing provider, ensuring that the data that you actually obtain is of the highest standard and highest quality. So we've actually been present in Europe for over 10 years now, with our newest lab open to you as of this year in Munich. Yeah, so for those who already been working with us, we've been probably accepting your samples, and you've been sending your samples to UK Cambridge. However, now it's become a lot easier where you can actually send your samples straight to Munich in our German lab. And if you're ever in Munich by any chance and would like to see the lab, we will happily accommodate you guys and give you a tour with our lab manager. And you can actually see the whole 1,000 square meter space of lab and office. And please let us know, however, 24 hours in advance. So as an update, we actually have 2 NovaSeq X Pluses in Munich, and they're up and running with the capacity to sequence 32 terabytes per day. We also have 2 of the latest long read sequencers, PacBio Revio, which can sequence 720 gigabytes of hi-fi reads per day. So we don't actually have an Oxford nanopore in Munich at the moment, but it is available to you guys in our other labs. So our other labs have actually a patented robot, which is called Falcon 2, which is an automated robot that allows us to have your samples sequenced from end to end with library preparation and everything installed. It is also planned to be installed in our Munich lab this year, so very, very soon. So if you are in Munich, you probably might see it already. And also feel free to watch the video that we have on our website where our Falcon is in action, moving around through the lab. It's really, really cool and quite awesome to watch. So of course, we have to ensure that your data is protected on our large servers, and we're planning to actually even upgrade our server in Munich. So also for you to know, if you are sequencing with us and have your data then provided from us, we don't actually store your data long-term. So 30 days after your project has been completed, it will actually be completely deleted. So please, please, please download the data and store it on a hard drive because we won't be able to recover it once we've deleted it. Yeah. As a brief overview, as you can see, we can and have sequenced samples of all areas of research, anything from food safety, agriculture, infectious disease, industrial diagnostics, we also do, and also museum samples. So a huge range of things. So anything that you are unsure of if we can sequence, please reach out to us and we can let you know because it's most likely that we can. Also, as a brief overview of the range and various sequencing services that we have available to you, with our latest here in blue, which is the spatial transcriptomics, more for human and mouse. And then we have on the epigenetics chain, we have enzymatic methylation sequencing, which is a less harsh alternative approach in comparison to whole genome bisulfite sequencing, which Arman will touch on further. So as I previously mentioned, we deliver high quality data and do have high standards, but we do deliver these high quality data consistently. And no matter what size your project is or how many samples you have, our expert team can really most definitely accommodate that. For example, we've just recently obtained more than 1,000 samples from a biobank and it's currently running and up and going. So, if you have a larger sample size, don't feel stressed, we can handle it. We also ensure that you have the golden standard and of course the newest technology for your sequencing projects. So if you're ever unsure which services you need or you're not sure how to start your project, please feel free to let us know. You will have a whole team behind you. And in case you have also DNA and RNA extraction problems, feel free to reach out. We will help with your protocols or optimizing them. And also even after your project is completed, if you have some bioinformatic analysis questions, feel free to reach out. So actually you don't have to feel alone when you're pushing the boundaries of science. For those who have not yet had a project with us, I briefly want to touch on our workflow. So initially we have a personal one-to-one discussion about your project and your aims and what you'd like to achieve. And once we have established that, a quotation will be sent out and we will require you to sign it or send a PO number back to us. After this, you'll be contacted by your technical specialist who will then help you with your sample information, preparing your samples for shipment, etc. And we will ask you to upload the sample for information form to your customer service system, which is an interface of Novogene online and where you can actually track your project throughout. And then at this point, we will also ask you to share your tracking number in case you don't choose our shipping pickup service. And then from this point on, once it reaches our lab, we will let you know, and we will also do the sample quality control. Here, we actually require you to confirm and select which samples you want to continue with. In case you only want to do a pilot study, for example, and continue with one, we can hold the rest of your samples for you, and then we can continue that later if you want to review it. So after this confirmation, we will then continue on to library preparation, sequencing, and bioinformatic analysis. If you chose bioinformatic analysis, we will ask you to actually fill out a form so that you can select which samples you want to compare with which. And then after sequencing is complete, it will automatically start. After the project has been finished and everything is done, you will receive a settlement where you have a final report, a statement, where you pretty much have to confirm that it is your project. You will get like a little tiny demo of your results, and you can then say, yes, this is my samples, please release the data to me. And once the payment is made, the data will then be released to you, which is why we actually send the invoice a little earlier, around after sample quality control, so that it can be processed at the same time in parallel by your purchasing department, so that as soon as this project is complete, you will get your data. So yeah, if you have any concerns, you can always reach out to me or your respective account manager who I can direct you to. But now I would like to hand it over to Arman, who will dive into the details of next generation sequencing and what we can actually offer.
Thank you for joining us for today's webinar. Today, I will start with the definition of nucleic acid sequencing, followed by talking about some of the current sequencing technologies and platforms. Then I will discuss some sequencing applications. Finally, I will talk about the general sequencing workflow that we follow here at Novogene. So starting with the definition, DNA sequencing or RNA sequencing is the process of determining the nucleic acid sequence, that is the order of nucleotides in the DNA or RNA molecule. Now, this sequence of nucleotides, it encodes the biological information that cells use to develop and perform their function. Thus, understanding the DNA sequence is fundamental in order to understand the function of genes and the genome overall. The pictures you see here show the many species Novogene has worked with for sequencing projects, and our experience ranges from human samples to plants to animals and also microbial species. Now let us look at some of the milestones in genomic sequencing. The first draft of human genome was published back in 2001. And since then, there has been a lot of advancement in this field, which is very nicely summarized in this publication that you can go through. And over the years, we've seen advancements in sequencing technologies and platforms and applications. Today, we are going to discuss some of them. And I will start with the evolution of sequencing tools. So broadly, we have 3 generations of sequencing tools to date. By first generation sequencing, we refer to, for example, Sanger sequencing, which is a low throughput method and is able to sequence fragments as long as 500 base pairs to about 1,000 base pairs. And I'm sure most of you in the audience would have probably used this method already in your projects. Then we have second generation sequencing, which is basically high throughput sequencing. And it is mainly represented by the Illumina platform. Now, this technology generates high throughput data, and it has revolutionized the field. And you can usually sequence fragments between 50 base pairs to about 500 base pairs in length. Now, both first generation and second generation sequencing platforms, they're considered to be short read sequencing platforms. Finally, we have third generation sequencing platforms, which are represented by Illumina, which are represented by PacBio and Oxford Nanopore Technologies. And these platforms allow us to do long read sequencing. That is, we can sequence tens of KBs of fragments on average. Another special feature is that both of these platforms can sequence native DNA or RNA in real time with single molecule resolution. And so when we talk about next generation sequencing or NGS, a lot of people associate it only with Illumina or second generation sequencing technologies. But often this term is used broadly to also include third generation sequencing. Now I will talk about the first generation sequencing and third generation sequencing platforms in more depth. So first generation sequencing is represented, for example, by Illumina sequencing. And the workflow here starts with the library preparation, wherein the DNA, it is broken down or fragmented into smaller pieces, and it is ligated to various adapters with the aim of creating a DNA library. Afterwards, the DNA library is loaded on to the machine, where the DNA library molecules, they hybridize to the flow cell. And then following several amplification cycles, we get these amplified clusters on the flow cell. Then we carry out sequencing. And during sequencing, several fluorescently labeled nucleotides are incorporated through several sequencing cycles. And the machine records the fluorescence and turns it into a DNA sequence. Illumina sequencing has several advantages. Firstly, it generates more data with the same DNA quantity in comparison to Sanger sequencing. Then it has a very high throughput because millions of sequencing reactions can run in parallel. Also, Illumina has been used widely in the field. So we have several applications for it. We have several analysis pipelines developed for Illumina-based sequencing. In terms of disadvantages, so it's a short-read sequencing platform. So you can sequence usually maximum up to 500 base pairs, more or less. And then compared to Sanger sequencing, it is more expensive for low target number. So if you're only interested in, let's say, one gene or one small plasmid, then maybe Sanger sequencing would be a cheaper alternative. So here you see images of NovaSeq 6000 and NovaSeq X Plus. These are Illumina platforms. NovaSeq X Plus is their latest platform. And Novogene is happy to actually provide sequencing on both of these platforms. Now I will talk about third generation sequencing, and it is represented by platforms from PacBio, like PacBio Revio, and from platforms from Oxford Nanopore Technologies, like the PromethION platform. So starting with PacBio. So the first step here is also preparation of a library. So we ligate adapters called SmartPel adapters to the DNA molecule. And after that, this library is loaded on to the machine, where a DNA polymerase that is fixed at the bottom of the well, it starts sequencing the DNA fragment using several fluorescently labeled dNTPs. Now, as the synthesis proceeds, the machine detects the fluorescence and assigns the nucleotide sequence. So this technology is actually quite similar to the Illumina technology in the sense that we are using these fluorescently labeled dNTPs, and this DNA replication reaction is going on. One of the advantages of PacBio is the possibility of these two sequencing modes. So we have CLR mode, or continuous long reads mode, and we have another mode called CCS, that is circular consensus sequencing. So in CLR mode, each DNA fragment is sequenced only once. An advantage of this is that it can sequence large insert fragment libraries, and it's often used for de novo assembly. The disadvantage is the relatively high error rate, because each DNA fragment is only sequenced once. On the other hand, we have the CCS mode. Here, each DNA fragment is sequenced multiple times, which means that any base errors that maybe came during the first pass or the second pass will be corrected in the subsequent passes or in the subsequent readings. So it provides us with highly accurate reads called hi-fi reads. And the latest platform from PacBio, which is the Revio, it delivers these hi-fi reads or this hi-fi data by default. So the advantages of PacBio platforms include long reads. So it's a long read sequencing platform. And then it is highly accurate with CCS mode. And also it allows direct detection of epigenetic modifications on DNA. The disadvantages, well, it's relatively more expensive compared to the short read sequencing platforms. And it requires higher amount of starting material. So usually you require starting material in microgram magnitude. But yes, it's a long read sequencing platform. And the applications which particularly require long read sequencing, such as de novo assembly, or if you're trying to look at large structural variants, PacBio and Oxford Nanopore, they're really helpful for those kinds of applications. So coming to Oxford Nanopore, now the sequencing principle and the technology here is a bit different because it relies on the use of a nanopore, hence the name. So this nanopore is fixed on a membrane. And during sequencing, a single strand of this DNA, it passes through the nanopore from one side of the membrane to another. And with every nucleotide that is crossing the nanopore or that is passing through the nanopore, there is an accompanied change in the ionic current. And this change in the ionic current, basically this drop in the ionic current, can be recorded to identify the nucleotide base. The advantages here, well, again, it's a long read sequencing platform, but you can get longer reads compared to PacBio. So I think Oxford Nanopore has the world record for the longest nucleic acid fragment sequenced. Another advantage is that there is a possibility of directly sequencing RNA, which was not offered in an Illumina or PacBio. And also this means that there is a possibility of direct detection of RNA modifications. Disadvantage, well, again, here you require higher amounts of starting material. And the base calling accuracy is lower in comparison to the other platforms that we've discussed, but the data can be improved through computing methods. An example of a platform from Oxford Nanopore is PromethION. And at Novogene, we do offer sequencing on the PromethION platform. So if we have to look at all these platforms together and compare the advantages and disadvantages. So looking at Illumina, the advantage is that it's a high throughput sequencing platform. It's cost effective, it has low error rate, and it is used widely in the NGS industry. The disadvantage is that the read lengths are short. On the other hand, if we talk about PacBio and Oxford Nanopore technologies, then these are long read sequencing platforms. And also they allow direct detection of epigenetic modifications. Also, these platforms are very good for sequences containing a lot of repeats. For example, centromeres, and also where the GC content is biased towards one direction. So for those kinds of sequences also, PacBio and Oxford Nanopore technologies are quite useful. The disadvantage here is that the costs are relatively higher compared to Illumina, and it requires higher amounts of starting material. Now let us look at some sequencing applications. Now at Novogene, we offer many sequencing services, and it won't be possible to cover all of them because of the shortage of time. So I will cover some of the sequencing services, looking at their principles, definitions, and so on. I'll start with DNA sequencing, and here I will talk about whole genome sequencing. So this is essentially a technique for sequencing the entire genome of an organism at a single time. And it provides us with a deep insight into the DNA sequence of living organisms. And it's also very useful because it helps us to identify and analyze various variants of the genome. So whole genome sequencing can be carried out with two different approaches. That is the de novo sequencing and the resequencing approach. So in the de novo sequencing, in this approach, we aim to assemble a reference genome for a specific living organism or a species for which the reference genome does not exist. So what we do here is that the genome, it is fragmented into smaller pieces, and later on, we create the library, and thousands of different sequencing reads are generated when we sequence the library. And during the data analysis stage, we assemble these sequencing reads in order to generate a reference genome that did not exist before. Now, with the resequencing approach, we aim to sequence a new individual within a given species, in order to understand, or in order to compare the differences between the genome of interest or the individual of interest and the reference genome that already exists. So here again, We take the DNA, we create the DNA library, sequencing is carried out, and the sequencing reads, they're aligned with the reference genome to compare the differences. So whole genome sequencing is beneficial because it helps us to look at various variants. For example, we can look at single nucleotide variants or indels or duplications, inversions. So all of these different kinds of variants can be detected with whole genome sequencing. Now, another point I would like to mention here is that if you're interested in large structural variants, then long-read sequencing platforms are better. Although for all these different variants, you can detect them through Illumina as well. But for these large variants, large indels, large copy number variants, long-read sequencing platforms might be more suitable. So I just covered whole genome sequencing. And I told you that you can do it with both de novo approach and resequencing approach. But we also offer whole exome sequencing. I don't have the time to cover the other applications. So now I will move on to epigenome sequencing. And here I will basically talk about DNA methylation sequencing. So epigenetics is essentially the study of how organisms control gene activity without changing the DNA sequence. Thus, these epigenetic mechanisms, they allow organisms to respond to their environment by altering phenotypes without a change in genotype. These epigenetic modifications can be inherited or even reversed. And epigenetic processes such as DNA modifications, chromatin modifications, and others, they play a role in several biological processes. So this gives you an overview of Novogene's epigenetics services. Today, I will just focus on DNA methylation and primarily EMseq or enzymatic methyl sequencing, because this is our latest offering. But we also offer ChIP-seq, RIP-seq, ATAC-seq, and others. And for more information, you can reach out to us. So DNA methylation, it is a process by which methyl groups are added to the DNA molecule. Now, usually it's the adenines and cytosines that can be methylated. And if we look at eukaryotes, then cytosine methylation is widespread. And as far as mammals are concerned, DNA methylation predominantly occurs in CpG dinucleotides in the cytosine guanine dinucleotide context. In other organisms, such as plants, we also observe methylation in non-CpG contexts. And it depends on the context, but DNA methylation usually represses gene transcription. So why study DNA methylation? Well, it helps us to answer several different kinds of research questions. Plant and animal researchers can use it to study development, behavior, environmental resistance, and agronomic traits. Clinical researchers can use it to study tumors, molecular markers, and some disease mechanisms. So DNA methylation has many uses, has many applications. So this is a comparison of different DNA methylation sequencing technologies, which are enzymatic methyl-seq, whole genome bisulfite sequencing, and reduced representation bisulfite sequencing. So I will talk about enzymatic methyl sequencing in more detail, but this is just to give you a brief overview of these three technologies. So enzymatic methyl sequencing, it uses enzymatic conversion to identify methylated cytosines, while bisulfite sequencing, which could be the whole genome bisulfite sequencing or reduced representation bisulfite sequencing, they use bisulfite conversion. So there is a harsh chemical treatment. In terms of genomic coverage, EM-seq and bisulfite sequencing, they cover the whole genome, while reduced representation bisulfite sequencing, it is for CpG-rich regions. In terms of damage to the DNA, because enzymatic method sequencing, it uses enzymatic reactions, so the DNA damage is minimal, while with bisulfite sequencing, the DNA damage is relatively higher. In terms of otherwise advantages, enzymatic method sequencing, it does less DNA damage. You can sequence samples with lower concentrations or lower amounts. On the other hand, bisulfite sequencing is considered to be the gold standard for methylation analysis. And RRBS is great because it's a targeted approach, and it's also it can be cost effective. It's great for pilot studies. So now I will move on to talk about EM-seq in more detail. So the EM-seq, this technology relies on the use of specific enzymes that can distinguish between methylated and unmethylated cytosine residues in the DNA sequence. So it uses two enzymatic steps with milder reaction conditions. So the first step is an oxidation reaction, wherein the TET2 enzyme and oxidation enhancer is used. So these reactions protect the methylated cytosines from deamination. Then in the second step, the DNA is made single-stranded and apobic enzyme is used. The apobic enzyme, it deaminates unmethylated cytosines to uracil, while these oxidized forms of methylated cytosines, they're not deaminated. Finally, we can do library preparation and sequencing. Now, this is a comparison of the two methods, that is bisulfite sequencing and EM-seq. And as you can see, both of these methods produce exactly the same sequence. The methylated cytosines are protected, while the unmethylated cytosines are converted to thymine. So what are the advantages of enzymatic method sequencing? Firstly, it minimizes damage to DNA. It produces higher quality libraries. Then it results in the same converted sequence as bisulfite sequencing. So as you can see here, that the final converted sequence is the same, which means that the same analysis pipelines can be used. EM-seq can be used for diverse sample types. It has uniformity of GC coverage, and also libraries show uniformity in coverage for all dinucleotides. Now, let's move on to RNA sequencing. So RNA sequencing is a sequencing technique which uses NGS to study or to reveal the presence and quantity of RNA in a sample at a given moment. So it helps us to analyze this continuously changing transcriptome. Now, in terms of applications, RNA-seq can give us qualitative data, which can help us to characterize, in short, what kind of transcripts are present in my sample, or to look at alternative splicing or orientation of transcripts and so on. On the other hand, RNA sequencing also gives us quantitative data or expression data. So we can look at gene expression levels. We can look at differential gene expression if we have a number of samples. And in general, RNA sequencing has many applications for discovering biomarkers, for studying host pathogen interactions, and so on. So there are many different kinds of RNA in the cells. We have, for example, messenger RNAs, which can translate into proteins. And in eukaryotes, they contain a poly-A tail. We can have different kinds of non-coding RNAs, like housekeeping non-coding RNAs, like ribosomal RNA, transfer RNA. Then we have regulatory RNAs, like circular RNAs or linear RNAs, which can also be further subdivided into long non-coding RNAs or small non-coding RNAs. And we are continuously discovering new RNA species. So depending on the RNA of interest, we have tailored solutions to sequence them. So for example, if you're interested in sequencing mRNAs, then if there are eukaryotic mRNAs, then we can offer poly-enrichment. If you're working with blood samples, we can do globin mRNA depletion. Similarly, for long non-coding RNAs, we do ribosomal RNA depletion. Again, if you're working with blood samples, we can do globin depletion as well. For small RNA sequencing, we perform size selection. For circular RNAs, we perform ribosomal RNA depletion along with linear RNA depletion. We can also do whole transcriptome sequencing to give you details of all of these different kinds of RNA species. And we also have solutions for ultra low input RNA. That means if you have low concentration of RNA, or we can also do exosome sequencing. The library type can be stranded or non-stranded. Now, let me tell you what I mean by this. So this is a comparison of stranded library and non-stranded library. So by standard library, we mean directional library. So here, we cannot find out the orientation of the original RNA transcript. Now, let me illustrate this with the diagram. So we have this mRNA here. And from this mRNA, we synthesize the first strand cDNA. Now, this first strand cDNA will be used to synthesize the second strand cDNA that you can see in green. Now, in a non-stranded library, when sequencing happens, the sequencing reads can come from either the purple strand or the green strand. However, when you carry out or when you use a strand-specific library or directional library, then during the second strand cDNA synthesis, we incorporate DUTP. And later on, we can actually digest this green strand here with an enzyme. So this DUTP strand can be digested. And all your sequencing reads are going to come from this purple strand, which was the first strand cDNA. Now, this can actually help you to decode the orientation of the original mRNA fragment or the RNA fragment. Now, this is useful because you preserve the information about the strand. You can identify antisense strand. It's better for defining the exact boundaries of adjacent genes. For a better quantification, again, this approach is more useful. And a lot of people who are working on epigenetics prefer to always go for a directional library. Now, finally, in terms of RNA sequencing, we can also do long-read RNA sequencing by using PacBio platforms and Oxford Nanopore technology platforms. Now, the advantages here include that because you have long reads, so it's great for studying the structure of RNAs. We can identify isoforms. We can more accurately detect alternative splicing events, and we can characterize novel genes and fusion transcripts in a better way. Also, we have a new advancement in PacBio, which is the SkinX kits. So these SkinX kits are based on the mass-seq method. So basically here we combine smaller amplicons into this large fragment, which allows us to increase the throughput, which also allows us to then be able to sequence these small fragments, which would have otherwise not been possible because PacBio is a long-read platform. And at Novogene, we are offering the option of using these SkinX kits. So please reach out to us if you're interested. Finally, I will summarize long-read sequencing and short-read RNA sequencing technologies. So with long-read RNA-seq, you can sequence long reads, so tens of kbs of fragment, while we can sequence fragments up to a few hundred base pairs. Then if you want to detect isoforms, then long-read sequencing technologies are more powerful. They're going to be better. In terms of accuracy, with PacBio, Hi-Fi reads, you have great accuracy, and Illumina has great accuracy. So mainly the difference is, I would say, in terms of the applications, because when you're doing long-read sequencing, it's great for annotation, for isoform discovery, for studying the structure of RNA. However, if you're interested in RNA expression, you want to do gene expression quantification, then short-read RNA sequencing, like Illumina sequencing, might be a better approach. Also, it is possible to do a combined approach or having a combined strategy that can actually utilize the strengths of both long-read RNA sequencing and short-read RNA sequencing. So finally, this slide gives you an overview of the different solutions that Novogene offers for RNA-seq. So we discussed some of them. So we offer mRNA sequencing and different kinds of non-coding RNA sequencing. But we also offer spatial transcriptomics that I'm not going to cover today. And finally, we also offer pre-made library sequencing. Finally, I will talk about microbial sequencing, mainly focusing on metagenomics. So let's start with some definitions. So what is a microbiome? So microbiome is a microbial community living in a particular environment. And cells from more than one species make a microbial community. There could be 2 microbial species, 3 microbial species, or hundreds of microbial species in a community. It's also important to note that a microbiome is a broad term. So it includes also community genes and gene products as well, which include RNA, proteins, and metabolites. So a microbial community may consist of bacteria, archaea, fungi, viruses, protozoans, and studying the microbiome is essential for research on humans, for animal research, agriculture research, environment science research, plant research. So it has a lot of applications. Also important to note is that if there are only cells of one microbial species, then it is a monoculture. So depending on whether you have a monoculture or a microbiome or a microbial community, the sequencing applications would change a bit, in the sense that if you have a pure culture or a monoculture, it's made of just one microbial species or strain. So you can isolate the DNA, and you can do DNA sequencing with either the de novo sequencing approach or the genome resequencing approach. Similarly, if you have RNA, you can carry out prokaryotic RNA sequencing. Now, when you have a microbial community, we can do what is known as metagenomics or metatranscriptomics. So if we have the DNA, we can carry out metagenomics with either the amplicon-based approach or the shotgun approach. For RNA, we can do metatranscriptomics. Also, all these different things can be done with both Illumina, that is short-read sequencing, as well as PacBio or Oxford Nanopore for long-read sequencing. Now let's start with metagenome sequencing. So I won't talk about these because it is similar to what we discussed so far. This is microbial whole genome sequencing, and this is prokaryotic RNA sequencing. And here, basically, we do ribosomal RNA depletion for doing the library prep. And so I will focus mainly on metagenomics and metatranscriptomics. So in metagenomics or metagenome sequencing, we carry out the genomic analysis of microbes by direct extraction and cloning of DNA from a microbial community. So What we mean by this is that you don't have to culture microbes. You can get a culture independent insight of microbial communities. So Novogene also, we receive samples from, let's say, from rivers or from the ocean, or you could have soil samples or stool samples. So samples can come from, many different backgrounds, and they can be then sequenced. We don't have to culture them in the lab first. So this is a big advantage of metagenome sequencing. So there are two approaches of metagenomics. One is a targeted approach, which is called targeted metagenomics or amplicon-based metagenomics or sequence-based approach. Another approach is the whole metagenomics, which includes shotgun and shallow shotgun whole metagenome sequencing. So in the targeted approach, we only sequence a selected set of marker genes that we think are present within the microbial community. On the other hand, the second approach is more global and is called the shotgun whole metagenome sequencing. In this approach, we sequence everything present within the microbial community. Now, shotgun metagenomic sequencing carried out at a lower depth is called shallow shotgun metagenome sequencing. So let's talk about this targeted approach in a little more detail. So just to give you an example of amplicon-based sequencing or targeted approach, we can actually sequence 16S or 18S ribosomal DNA, or in the case of fungi, we can do ITS sequencing. So we focus on just one gene and usually certain regions of the gene. And the advantage here is that it is cost efficient, it is fast, and it's quite a robust approach to study the microbial community. It's very widely used, and it's quite widely used in taxonomic studies and phylogenetic studies. And if you want information on the primers that we use for amplicon-based metagenomics at Novogene, then you can visit this particular link. On the other hand, when we talk about whole metagenomics or shotgun metagenomics, here we sequence everything which is present in the microbial community. So we are not biased towards a particular gene, but we take the sample from the microbial community, we extract all the DNA, and all of that is sequenced. So this gives us a comprehensive and unbiased view of the entire metagenome. And the popularity of shotgun metagenomics has been steadily increasing. And we see many publications coming up in the recent years. And it has many useful applications, such as studying community composition. And also you can do some amount of functional characterization with this approach. Also, again, I mentioned this previously. So we have the shotgun whole metagenome sequencing, but you can also carry out shotgun metagenomics at a lower depth. And when you carry this out at a lower depth, it's called shallow shotgun metagenome sequencing. Finally, again here, we have the possibility to do long read metagenomic sequencing. And there are several advantages of the long read approaches. So it provides us with comprehensive profiling, and you can study the true diversity of complex microbiomes with highly accurate species level resolution, and often you can reach up to strain level resolution or subspecies level resolution as well. Then there is enhanced shotgun metagenome assembly. So in case you want to assemble genomes, then with this long read approach, you can achieve, or you can assemble high quality MAGs, which are known as metagenome assembled genomes. Then also it provides us with precise taxonomic profiling. So if you do shotgun metagenomics with the long read approach, you can get exceptional taxonomic profiling data. Now, this is comparing amplicon-based approaches with shotgun-based approaches. Also, I've put here the long read amplicon-based approach. What is missing from the table is the long reads shotgun-based approach. So there isn't enough time to go through all of these parameters. But if we, let's say, talk about taxonomic resolution, then amplicon-based approach, that is the short read-based approach, it goes up to genus level. With shotgun-based approaches, we can go to species or even subspecies level with, for example, the shotgun metagenomics. If we do long reads, like long read-based amplicon, then again, we can reach species, subspecies level. And the same will also go for long read-based shotgun metagenomics. You can have a very high taxonomic resolution. In terms of taxonomic coverage, so depending on the gene you choose, it's going to be limited. For example, if you focus on 16S, you can look at, for example, bacteria and archaea, while shotgun-based approaches, they cover all taxa. So viruses, bacteria, fungi, everything. Now, functional profiling. Now, here in amplicon-based approaches, you're focusing on one gene. So functional profiling is not possible. Yes, you can extract some data, but if you're really interested in functional profiling, then shotgun metagenomics is the way to go, whether it is long reads or short reads. Now, another important point is sensitivity to host DNA contamination. Now, amplicon-based approaches, they're less sensitive to host contamination because you're focusing on a particular gene. While with shotgun-based approaches, we sequence everything. So, which means that you might also get a lot of host contamination. So let's say if you are taking up, let's say, samples from the human gut. So you will get microbial DNA, but you will also get a lot of reads coming from the human host. Similarly, let's say for plants. So this is an issue with shotgun metagenomics. In some cases, at the experimental stage, while you're extracting the DNA, you can try to remove the host DNA. Or otherwise, what we can do is we can try to sequence at a greater depth so that we can get enough of our microbial reads, and we can remove the host reads from our results. Now, in terms of overall bias, in amplicon-based approaches, because we are focusing on a particular gene, this is a biased approach, while shotgun-based approaches, they are unbiased approaches or untargeted approaches. Now, finally, for microbes, let's come to RNA sequencing and metatranscriptomic sequencing in particular. So it's an application, a specialized application of RNA sequencing that involves the analysis of total RNA transcripts, which are present in a complex microbial community, such as, again, those found in environmental samples or human gut, soil, or aquatic ecosystems. This technique allows researchers to study the gene expression and functional activities of multiple microbes within a mixed population simultaneously. And a subset of this metatranscriptome sequencing is dual RNA-seq. So here, instead of focusing on the community, many microbial species, we are focusing on two microbial species. So it's called dual RNA-seq, also sometimes known as simultaneous RNA-seq. So in this technique, we analyze the transcriptomes of, let's say, a host organism and a pathogen. simultaneously in a mixed biological sample. So it can be used to, let's say, study host pathogen interactions or some kind of symbiosis between, let's say, two species. So here, the preparation of the sample of the library, it depends on what kind of host and pathogen or what kind of species are we dealing with. So we could maybe carry out double ribosomal RNA depletion, or there might be poly-A capture if, let's say, it's a eukaryotic host species. So depending on that, we will process the sample. Now, finally, I will talk about the general sequencing workflow. So I only discussed few applications that we offer at Novogene. It's not possible to cover all of them in a short span. So now I will move on to another key point, which is the sequencing workflow. So the sequencing workflow essentially starts with experimental design. Now, this is usually done by the clients. And at Novogene, we do provide assistance in terms of experimental design, because this part also involves thinking about the number of replicates to do, or which sequencing platform should be chosen, what should be the appropriate data amount, which controls need to be included, what should be the depth, all these kinds of considerations. So all this comes during the experimental design stage, and then comes sample extraction. So extraction of DNA or RNA, which is done by the clients. And also Novogene, we offer some of our extraction services. So we can also do sample extraction. Now, after sample extraction comes sample QC. So at Novogene, we do sample QC, whether we extract the DNA or RNA ourselves, or whether you send us the sample, we do the sample QC at Novogene. So here, we first do gel electrophoresis to look at the integrity of the DNA or RNA. So we want to see if the bands are of the right size, there are no smears, there's no contamination, the bands, they look as expected. So we first do this gel electrophoresis. We also look at the profile of the nucleic acid on a fragment analyzer. So for example, in the case of RNA, the fragment analyzer can give us the profile, it can give us the RNA integrity number or RIN, that can tell us about the quality of the sample. And finally, we also measure the concentration of the sample using a qubit fluorometer. You can also measure it on nanodrop, but we recommend using the qubit. So after the sample passes the sample QC, we go ahead with library preparation. So during library preparation, we fragment the DNA. So this is the library preparation for Illumina. For Oxford Nanopore and PacBio, it's going to be different. What you see in the diagram here is the Illumina library preparation. So the DNA, it is fragmented into fragments of about 350 base pairs approximately. And fragmentation is then followed by end repairing and A-tailing. So this is important. This end repairing and A-tailing is important for these sequences to bind to the adapters. So because in the next step, we will add adapters for sequencing. So we add P5, P7 oligos, which help the DNA to bind to the sequencer. We add sequencing primers, indexes, and all of these then are, they're added to the DNA molecule to prepare a library. And finally, when the library is prepared, we do the library QC. I will come to the structure of the library in another slide. So after the library is prepared, the library QC is done, and the samples are okay. We carry out the sequencing. And for sequencing, we can have different strategies. Now, the most commonly used strategy at Novogene is paired N150. Now, this is compatible with most service types and can be used for whole genome sequencing, RNA sequencing, metagenomics, and it's also the most cost-effective strategy that we offer at Novogene. Then there's the possibility to do paired N250. This is great for short amplicon sequencing. We also offer paired N50 strategy for some short inserts or for ATAC sequencing. We also offer single N50 strategy, which is great for short RNAs or short inserts, or also sometimes for ChIP-seq and RIP-seq samples. Now let's look at the structure of a library. Now this is a structure of an Illumina compatible library. I don't have the time to go through the libraries for PacBio and Nanopore. I will mainly focus on Illumina. Now, it's really important to study the structure of the library because at Novogene, we also accept pre-made libraries. So you can also prepare libraries yourself and send them to us. But what is important is that the library should have all the key components. So here in the middle, you see the DNA insert. Now, for the library, we need to attach these adapters like P5 and P7. Now, these adapters are important because they help the library to bind to the flow cell. Then we add indexes. Now, these are important for demultiplexing. So indexes are usually between 6 to 10 base pairs in length. And then we have the sequencing primer, which should also be an Illumina compatible sequencing primer. So all these three components are crucial. Now, in terms of this index, we always recommend a dual index library. And also at Novogene, by default, we use a dual index library that eases demultiplexing. And in case there is some index hopping or something, there's always the other index that can be sequenced, and the demultiplexing can be done with accuracy. Finally, once sequencing has been done, we get our sequencing data and we do a data quality control. Now, in the data quality control, we do data filtering, which means we filter out bad reads, which might be reads that have low quality, or reads that might have some adapter-related contamination. We also look at the error rate of sequencing, and we also look at this ATGC content. And this data quality control report is also then shared with the clients. Now, if the data is of good quality, we then proceed towards bioinformatics analysis. Now, at Novogene, we offer bioinformatics pipelines for almost all of our applications. So I talked about whole genome sequencing, methylation sequencing, RNA sequencing, and we have tailored bioinformatics pipelines for all of them. Now, it's not possible for me to discuss all those pipelines within this short duration. So I'm just giving you an example of a pipeline for mRNA sequencing. So actually also for mRNA sequencing, we have multiple pipelines based on what you're interested in. So this is one of our standard pipelines. So we get the raw data from sequencing, we do the data quality control, as I mentioned, we map the reads to the reference genome, then we can do gene expression quantification, differential gene expression analysis, and finally some functional analysis. And we actually give you a bioinformatics report. So you can reach out to us for looking at a demo report. So this is an image from one of our demo reports. So if you decide to do bioinformatics analysis with us, we will give you a report with publication-ready figures and analysis. Also, if you are doing mRNA sequencing with us, Then we also offer you a cloud-based analysis platform, which is Novogene. So then you can also do some analysis by yourself online. We have a tutorial for you, and you can, maybe rearrange some groups and generate some figures for yourself. So with this, I would like to end today's webinar. I know there was a lot it was a lot that I covered very quickly. I did not have so much time, and there were many things, so I went by quite quickly, and I apologize for that. But if you have any questions, or if you need any further details, please reach out to me and Melina, and we will be very happy to answer your questions. Thank you once again.
Another question is about the conduct for troubleshooting regarding already with DNA or RNA isolation, if it's the regional representative or maybe you have a specific central troubleshooting team.
Yeah, so you would contact your account manager for that. If you have issues with DNA and RNA extraction, we will then, together with you and a technical specialist, go through your protocol. We can help you optimize it, maybe give you some tips on where it could be maybe crucial. Otherwise, we can also go through the kits that you're using and see if that has maybe something to do with it, or the species that you're working with, or how the extraction is actually being done. And if you want, then we can, yeah, of course, do this in an online meeting or in person, depending on who your account manager is. Yeah.
So I have some questions. Is there a possibility that somebody prepares the library in the lab and send the library for sequencing? If it's something that it's very specific, do you provide the kit for the library preparation, right?
Yeah. So actually, you can send us pre-made libraries for sequencing. But the key here, like I mentioned, is that those libraries should be compatible to our sequencing platforms. So that is why before you start preparing the libraries, it's actually better that you have a conversation with us, and then we can go through your kit, we can evaluate the kit, and we can check if it's compatible with our platforms. And if it's all fine, then you can prepare the libraries and we can sequence them for you.
Okay. Thank you very much, both of you. Thank Novogene for this wonderful webinar. I hope everybody enjoyed. I'm sure everybody enjoyed. We will upload this into our YouTube channel. And thank you both. And if you have any questions, please contact Novogene.
No problem.
That's what I do.
It's been a pleasure. It's really been a pleasure to be here with you guys. And also just the last slide for the Recrop members, we have a Recrop discount. Therefore, 20% will be automatically given to you. And this will be valid until December 31st. So until the end of the year. And just feel free to submit that to me per email. Or you can also ask Akis if you don't have my email, otherwise it's listed here. And then I will direct you to your responsible account manager for your region, since we're all over Europe. And yeah, I'm responsible for Germany. So Frankfurt, for example, feel free to reach out. But otherwise, the 20% discount is applied for UL.